Hi, my name is Derek Reiman, and I'm a PhD candidate at the University of Illinois at Chicago under Professor Yang Dai. In this presentation, I will be talking about some of our work using conditional generative adversarial networks to boost the performance of machine learning in microbiome datasets. In our work, we will be looking at microbiome data. The microbiome consists of microscopic organisms cohabitating in a shared environment, and it has been shown to have a significant impact on both host development as well as the pathogenesis of various diseases. These organisms have been shown to have a profound impact on its environment. Of particular interest is the human microbiome and how its composition can affect the health and development of the host. Recently, machine learning models have been utilized for the prediction of host phenotype. However, microbiome data often has far fewer samples than features, which can quickly lead to the overfitting of models. To address this challenge, we propose a novel method for generating synthetic microbiome data using conditional generative adversarial networks, or CGANs. We then use the CGAN-generated synthetic samples to improve the performance of machine learning models for prediction of host disease status. Conditional Generative Adversarial Networks, or CGANs, are a data-driven non-parametric way to sample data points using two competing networks. The first network is called the generator, and it is shown in the image as G. This network is tasked with generating a microbiome sample, which in our study will be a set of microbial abundance values. It generates the sample using a vector of random prior values, shown as Z, as well as additional site information shown as S. In our study, this site information will be the disease status of a patient from which the sample was taken. The second network is called the discriminator, and it is represented as D in the image. The discriminator is tasked with determining if a given sample is a true observed sample, or if it is a synthetic sample generated by the generator network. To make its prediction, it is given a set of microbial abundances as the sample, in addition to the site information, representing the disease status of the sample. These networks are trained iteratively, such that the generator network tries to trick the discriminator network, as the discriminator learns how to discern between the real and synthetic data. This type of setup is similar to a min and max game, and can be trained to Nash equilibrium, at which point the generator network creates synthetic data that closely follows the distribution of the real data, and the discriminator network predicts at 50%. Once trained, the generator network can then be used to generate large sets of synthetic data. In our study, we will use two datasets of patients with inflammatory bowel disease, or IBD. The first dataset is from the Prospective Registry and IBD Study at Massachusetts General Hospital, which we will call PRISM. Additionally, we used a second IBD dataset consisting of a combination of two independent cohorts from the Netherlands for external validation. Microbial and metabolite features were obtained from the study in the form of relative abundance. Models were trained using 10-fold cross-validation. This means that the data was partitioned into 10 partitions, and for each partition, 90% was selected to train the model and 10% was held out to test the model. CGAN models were then trained for 25,000 iterations, where 64 samples were randomly selected for updating the models in each iteration. Every 500 iteration, the model was stored and a visual analysis was performed. This analysis included the principal coordinate analysis of the training set, the principal coordinate analysis of a generated set using the site information labels of the training set, and finally, the principal coordinate analysis performed on the combination of the two. We then selected the best CGAN model by visually inspecting the plots and selected the model which provided the best overlap. That is, the real and synthetic data were indistinguishable. We then used this model to generate 20,000 samples, 10,000 of which were IBD samples, and 10,000 of which were healthy samples. Here is an example of what the visual plots of an optimal model were. On the left, we see the principal coordinate analysis of the training set used to train the model. In the middle, we see the principal coordinate analysis of the synthetic dataset that were generated using the same site information provided by the training set. And lastly, on the right, we see the plot of the principal coordinate analysis of the joint dataset. As you can see, this model was selected because not only did the real and synthetic data follow similar patterns, but when combined, they overlap and separate based on disease status rather than if they are real or synthetic. To further see how well the synthetic data samples followed the distribution of the real data, we compared alpha and beta diversity metrics. Diversity metrics are commonly used in microbiome studies to describe the richness and diversity within and between samples. Alpha diversity is used to describe the diversity within a single sample, and in our analysis we use Shannon's entropy as shown in the equation here. Beta diversity, on the other hand, represents the diversity between two samples. For this, we use the Bray-Curtis dissimilarity, as shown in the equation on the right. In this equation, CAB represents the minimum value of each feature between samples A and B. SA and SB represent the total sum of features within samples A and B respectively, and since we are using relative abundance, this sum is equal to 1. The distribution of alpha and beta diversities are shown in the bottom of the center column. Here, the blue represents the distribution in the true data, and the orange is the distribution in the synthetic data. We also represent the value of randomness by calculating the distribution of diversity measures using samples from the randomly initialized CGAN before any training. What we observe is that in both alpha and beta diversity, by training the CGAN model, the distribution of our synthetic data shifts from the green to the orange, and the orange is closely following the pattern of the blue distribution, showing that we are learning how to model the microbiome data. Next, we train logistic regression and neural network models to predict the disease status. We train models using the original data, as well as models augmented with synthetic data. We then evaluate these models using the 10% held out test set from the cross-validation partitioning. And since we are doing 10-fold cross-validation, we can do this 10 times to get a fair evaluation of performance. We evaluated the models based on the area under the receiver operating characteristic curve, or ROC-AUC for short, and we observed that in both logistic regression and the neural network models, there was a significant increase in performance by using the synthetic data. And here significance was calculated using the Wilcoxon sign rank test on paired partitions. Lastly, we performed the same experiments, but this time we used the entire PRISM dataset to train the CGAN. We then evaluated how will it generalize to the external IBD dataset. We observed that the joint principal coordinate analysis of the PRISM synthetic data using PRISM's labels and the external set overlapped well when using the best selected model. In addition, we see that the distribution of the diversity measures in the external set follows similar distributions to the synthetic data as well. This is important because it shows that the CGAN did not overfit to the PRISM data. Then we performed the same prediction analysis using logistic regression and multi-layer perceptron neural networks, and found that using the synthetic data improved the performance again. This shows that the synthetic data generated from the CGAN not only augmented the dataset, but was also able to be transferred to a similar external dataset. Thank you for listening, and I hope you enjoyed the presentation. If you have any questions, feel free to reach out to either myself or Professor Yang Dai. Thank you again.